Thank you very much for being here today. As when you look around a little bit. Look around at people that you know, look around at people that you don't know. My profound gratitude to everyone who is here. It goes the other way around, that I think it's a moment to reflect on how unusual, in a good way, gatherings of this intensity, magnitude, and purpose are in our personal and professional lives. So I'd like to welcome everyone to the 5th annual Rare Disease Symposium, and there are many people who contributed. Many of them are sitting around this table in front in a sort of organizational way. But what is more important, I think, are the people who contributed around these tables, or perhaps not present today, but yet present in extension of their work with generosity of their mind and their goodwill to participate in something that has been either misunderstood or overlooked or just dissipated in parts of the business that healthcare is today in this country and research is today in this country. And I'm not being critical. I am just stating the obvious. The first mistake, perhaps, is that that whole concept of rare disease. The rare disease, in my opinion, doesn't exist. The rare has been, if it had any functionality, anytime, anywhere, it was a tool, it was a heuristic, it was a lever to get funding for things that otherwise would not be funded. But the whole concept of rare is erroneous on several different levels. First of all, there are about 30 million people that have so-called rare diseases on this side of the Atlantic and similar number on the other side of the Atlantic. So this is as common collectively as many, many other conditions that we consider very common. The second reason why it is not rare is that when we look at all of these common diseases, and my clinical practice is in bone marrow transplant, so cancer comes to mind, but cardiovascular, strokes, and others can be considered, diabetes, obesity can be considered in the same category. We have learned very well over the last 2 decades or so with mapping of the human genome and epigenome and other layers of regulation on the molecular and cellular level, that the personal contribution of the genotype, phenotype, environment that converge on the disease manifestation that you can see in your clinic or in your loved ones is unique in everyone. Every cancer I've ever seen and treated is unique and has to be considered in its uniqueness, because that's where the now almost cliche term of personalized medicine comes in. I prefer the term high definition medicine because it brings in the acuity of the information and the data that we need, not just from within the cells, from the digital code of the genome, but also from the environment, the burden of disease, the disease cascade, and all of these other relevant aspects of view. And then the last one would be, if it is you or your loved ones, is definitely not rare, because the horizons of your life are typically filled with that concept, and from the personal, humanistic, and profoundly felt on a personal level point of view, there is no such thing as a rare disorder. And we all know that a lot of things happen in our genetic and clinical, as long as I'm going to focus on the disease aspects of this life, that are almost like fate. They are probabilistic, obviously, but they are indeterminate, undifferentiated in their impacts, and that's what makes it also resonant, with this group, and creates, I think, the tremendous momentum and deference that this idea of a Rare Disease Day and Rare Disease Symposium brings around, that everybody needs to be paid attention to. Now, almost 3 decades of my practice my clinical practice have been spent on what is called, erroneously as we established, a rare disorder. The science that has differentiated magic in medicine from medicine that you know has built a lot of power, a lot of scientific power around what we can do about this. So we no longer, in many of the conditions that are genetic perhaps, and occur by random and non-random insults in the genome, we have been able to understand them better. But when I look at the continuity from the information to something actually happening, it is really that we have the sciences, and I would argue strongly humanities and arts, that help us to understand the human condition, either in this molecular or its perceived way. But these are like Google Maps. These are like directions of travel. You know, this is how you can get to a certain position where you want to be. In this position, of course, it would be you would like to alleviate suffering of someone, or perhaps even effectively treat, or in some instances cure some of these conditions. But what is decisive, in my opinion, is not that Google map that science and arts built for us, or we build them, you build them. It's really the passion that comes, that you decide, I'm going to take this path on the Google map. I'm going to take this path, and I will be like a bird dog. I will not give up until I get there on that trail. That ingredient, that absolutely necessary ingredient, does not come from science alone, but comes from surroundings and gatherings, virtual or real like today, like this one. So I'm very grateful to everyone for being able to do this. And as we go through the day, from the very beginning to later on, it is really that you are making a lakeful of knowledge out of the spoonfuls that my grad students and others have brought to the poster session and to the discussions around this. And it is our consolidated, prolonged non-destruction that I think enables where we need to go. And the good news is that we have very powerful ambassadors, very powerful advocates. The president of the university, John Gable, is one of the people who have been who would have liked to be here and would be delighted to share with you, if she were here in person, what you're going to hear in a minute, which is her focus on quality, equality, well-being of everyone at the university, and also that magnificent power that the science and humanities at this university prepare as the substrate for these Google Maps that I described. So let's hear President Gable first.
Thank you to the University of Minnesota Medical School, the College of Pharmacy, the Center for Orphan Drug Research, and the Stem Cell Institute for sponsoring Rare Disease Day. There are more than 7,000 known rare diseases that affect approximately 30 million people in the United States. Across Minnesota, many families are dealing with the realities of these diseases every day. Approximately 50,000 Minnesotans have a rare disease, half of whom are children. The University of Minnesota is a leader in rare disease and orphan drug research, care, and education. Our medical school continues to lead the way, gaining in reputation for treating some of the most challenging cases that often would be turned away elsewhere. And our faculty physicians are among the most experienced in treating these devastating diseases. The College of Pharmacy and its Center for Orphan Drug Research have improved the care of individuals suffering from rare diseases through research, education, and public policy since 2005. The cutting-edge studies and clinical trials conducted by our faculty will produce new treatment options that will ultimately enable patients and their families to live fuller, healthier lives. With all of this amazing work going on here at the university, it was no surprise that last year, our Minnesota legislators requested that we establish a Rare Disease Advisory Council, consisting of professionals across the Minnesota healthcare community and people who have a rare disease themselves. I am so proud of the work you're doing and the commitment you've made to patients, families, and communities across our state. I encourage you to continue this great work, and I challenge you to find even better ways to connect patients and their families, healthcare providers and organizations, and industry and government leaders with rare disease-related clinical care, research, and education. Thank you for all you do to serve others and the University of Minnesota.
Very good. She's very good. She means it, right? So the other ambassador, the other supporter that you have, that I have, is the U.S. Senator Amy Klobuchar. She's wonderful too. I had an honor of meeting with her several times, and she's one of the, doesn't matter the political affiliation. I specialize in collecting good people, and she's one of the people who really mean to do good for the society in whatever our station is. So she's sending you a letter that I'm going to read for you. She says, "Dear friends, thank you for the invitation to join you at this year's University of Minnesota Rare Disease Day. I want everyone to know how thankful I am for the unrivaled dedication of the researchers, advocates, students, families, and patients here today. You all are leaders in the rare disease community, working every day to make breakthroughs that will help us better understand the human body, identify treatments and cures, and save lives. Throughout my time in the Senate," I'm still reading. Okay. "I have consistently pushed my colleagues to support the NIH and enhance medical research funding in budget and appropriations negotiations. But funding isn't the only answer. We must also continue elevating the needs of patients and their families and making sure they have a better seat at the table when the FDA is reviewing new treatments. Patients and families need greater clarity about the process for approving these drugs, and their input should be valued at every step of the way." Passing the 21st Century Cures Act just over 3 years ago was an important milestone for getting patient experience into drug approval process. But that's just the start. This is why I have continued working with Mississippi Senator Wicker to ensure the FDA consider patient experience or patient-focused drug development information as part of the risk-benefit assessment. Seems pretty obvious. Moving forward, there's still a lot of work to do, but I am working across the aisle because members of both parties know how important research is and how many families are counting on the ability to access the treatments and cures that are the result of the work going on at the NIH. I am confident that together we will accomplish some truly remarkable things. We will find treatments and cures, we will save lives, so thank you for everything you do for rare disease patients, and keep up the good work. Sincerely, Amy Klobuchar, United States Senator. So again, you know, people get it. People get that this is an important work, and people get that getting together in this fashion is essential for advancing, you know, where we need to be, where we are not yet, obviously, but where we need to be, I would hope, in several years or within a decade. Now, to sort of complete the trifecta of the phenomenal people that are surrounding, you know, this endeavor and this thought, and mind you, all three of them are women, I go to Erica Barnes. Erica Barnes, you know, is, you can finish chewing whatever you're chewing. I still have something to say. Is my friend. I have been profoundly impressed with her ability to deal with disorder that her daughter Chloe had, and how she transmuted the tragedy, the vicissitudes of life into something remarkably positive. I can tell you that there is no more dedicated, no more articulate, no more well-read she reads Marcus Aurelius and Kierkegaard. You know, we exchange books on that. Articulate ambassador for rare disease disorders. And as the president mentioned, she was the driving force behind the Rare Disease Council that has been established by the legislature this last year. And again, you know, one of the tireless individuals that we look up to, I look up to, and I'm very grateful for everything you do. Please welcome Erica Barnes.
Thank you, Dean Toller, for that introduction. And I just want to also thank Dr. Cloyd and all of the staff at the Center for Orphan Drug Research and all of the committee for planning this. I've been involved in Rare Disease Day now for about 3 years, and every year I'm just amazed at how the event grows and how much more participation that we have. I also want to thank all the researchers, industry, medical community, and future medical providers, students that are here today for attending the event as well. But most importantly, I want to thank all of you patient advocates who have come out today for Rare Disease Day to raise your voice and to be a part of this and to better understand what we're doing through the Rare Disease Advisory Council and at the University of Minnesota to address the needs of the rare community. So thank you all so much. So I'm here today to talk a little bit about the progress that we are making in carrying out the work of the Minnesota Rare Disease Advisory Council. As Dean Toller said, that was legislation that passed in the last legislative session, and I'm the administrator of the council, and the council is actually formally in the legislation called the Chloe Barnes Rare Disease Advisory Council, and as you can tell by the last name, it was my daughter. So it is very fitting that my presentation is going to coincide with a very timely and informative discussion around the FDA's efforts to balance efficacy and scientific rigor with flexibility, and that flexibility that is necessary to bring urgently needed treatments to the rare disease market. And one of the reasons that it's fitting is because the FDA in a certain way provides guidance to the state of Minnesota and provides guidance to our healthcare system as we seek to define and carry out all the work before us. And what I mean by that is the Federal Drug Administration is one of our most respected and established institutions, and it has long recognized the uniqueness of the rare disease journey in terms of drug development. Since 1983, with the passage of the Orphan Drug Act, and I don't know, Sarah, if you'll talk a little bit about the passage of the Orphan Drug Act, since that time, the FDA has developed a whole process for rare drug approvals. And so much so that when a company applies for orphan drug approval, they have a whole separate track that drug goes through. And so I've often thought that it stands to reason that if the FDA has a whole different set of approval processes for rare disease drug development, why has that not been applied to the patient journey in our healthcare system? So one of the visions that I have is that we would follow the lead of the FDA as a healthcare community and really understand that there needs to be processes in place to help patients, you know, and processes in place so that we're not just understood the way that someone with a more common disease is treated in our healthcare system. So that's a very driving passion for me and a very driving concept for me. The council's task is a large one, but it's also urgent and necessary. The urgent task of our council in the state of Minnesota is better integration of the rare community into care. And it's with this goal in mind that we as a council have spent the last few months really crafting a vision and a mission statement. We've spent the time since we convened in October thinking through what our mission and vision is. So I'm going to read for you the vision of the Minnesota Rare Disease Advisory Council. The Minnesota Rare Disease Advisory Council envisions a state where every Minnesota citizen living with a rare disease has access to a timely diagnosis, expert and coordinated care, as well as individualized treatment, management, and support throughout their lifetime. Obviously, that's a big vision, but it's what rare patients deserve, and it's what we're going to work toward as a council. But before I talk about the council priorities, I want to highlight the significant role that patients themselves played in creating this council. Some of you in this room were very intimately involved in the process of passing the bill for the Rare Disease Advisory Council, and some of you maybe aren't aware of the work that went on behind the scenes last year. So in 2017, a small group of patient advocates began meeting at the Southdale Library, some of you are in this room, to discuss the pressing needs of the rare disease community and how we could collectively raise our voice. While we all came with different diagnoses, we were struck with the realization that the same barriers to care in each of our communities were being replicated time and time again. We might have had separate diagnoses, but we had very common stories and very common experiences as we interacted with the healthcare system. A years-long delay in receiving diagnoses, lack of general practitioners in our local communities with any knowledge of our disease, specialists continents away or non-existent specialists, and the fact that only 5% of all the rare disease patient communities, roughly 5%, have any FDA-approved treatment. These were all things that made us realize that the root of the problem was one with the system, and we wanted to address it in a systematic way and push for systematic change. So we began to advocate for the creation of a Rare Disease Advisory Council that would convene all of the stakeholders in the state of Minnesota to more intentionally and systematically address the gaps in care in the rare disease community. If you read the legislation, there's a section that articulates a series of duties that the council is charged with, and as a council, we are very dedicated to enacting those duties and following the legislation in honoring, meeting the requests of the legislature. These duties are not the result of esoteric hypotheticals or just sitting around the table in a very esoteric way. All of the duties that are laid out in the legislation were actually forged by the life experiences of real-life patients in the state of Minnesota. And again, I know some of you are in this room. Some of these patient advocates testified before committees and told about their journey with rare diseases. For example, the charge to provide education to reduce time to diagnosis was included because there's a mom in Minnesota who waited 5 of her 6 years of her son's life to find out what was wrong with her son. So for 5 years of his life, she had no idea what was causing his symptoms, and she testified about the journey of a mom who has to wait that long for a diagnosis. The duty to increase awareness for the need of more treatments is in the bill because a father and a mother with no medical background have had to conduct their own research before being able to convince the medical community to pursue a possible treatment for their 2-year-old son. And that family actually couldn't be here today because their son has been sick the last couple of days. But their journey with not having an effective treatment for their son was a driving force behind making sure that we address the lack of treatments. The duty of identifying problems when switching health plans was included because a mother who changed jobs was told that her new insurance would no longer cover her son's life-saving medications. These are just a few of the stories highlighting how patients' experiences have influenced legislation. I'm sure there are many of you in this room that could tell the same stories and could tell other stories about your journey with rare diseases. So the bedrock of this council is the patient advocates and the work that they did to make addressing the needs of their needs a priority of our state. But patient advocates didn't do it alone. We couldn't do it without all of the stakeholders who also joined us. Care providers who tirelessly work in their free time to do extra research and write letters of medical necessity contacted their legislators, and I know you guys are also in this room too. Health system leadership who knows that the process needs to be improved wrote letters of support from their institutions. Industry partners who want to see innovation applied in the rare disease space lent their expertise. This was truly an organic and collaborative effort on the part of multiple stakeholders. But where are we now? I pulled up on the screen some of the pillars of the Rare Disease Advisory Council that we've identified. There's 24 members on the Rare Disease Advisory Council, and our task is to turn all of those aspirations for improved care into tangible goals. And we've been working for the last few months to identify those goals and priorities so that we can start to address them. So we have what we call 4 pillars of focus. And I'm going to go through each one of them right now and discuss them a little bit with you. So pillar number 1 focuses on a deeper understanding of the rare disease community. The statement that we have is that we will have a deep and comprehensive understanding of the systemic barriers across patient groups that are unique to the rare community. Due to the lack of collective consciousness historically around rare diseases as a group, there are many gaps in knowledge related to rare disease impact and care. Baseline data is often very scarce, and I'm sure I don't need to tell any of you who are a patient in this room that finding baseline data, protocols about your rare disease can be very, very difficult and very scarce. Even knowing the incidence and prevalence of rare diseases is sometimes a challenge. So, Additionally, there's no quantitative information on how many providers with expertise in rare disease are even practicing in the state of Minnesota. The basic fact is that metrics are very rare in the rare disease community. The Council ascribes to the axiom that what you cannot measure, you cannot improve. So we will take the time to gather data from patients and providers to determine what these groups believe are their biggest barriers to care, as well as regularly communicate with the patient populations in the state of Minnesota. Pillar number 2 states that the Rare Disease Advisory Council will develop recommendations and resources to improve access to and coordination of care for rare disease patients. There are many factors that contribute to the reduction of the coordination of care. And I'm sure, again, many of you could list many of the issues that you face with coordination of care, but one of the examples that has stood out to the Council is the fact that pediatric patients who live into adulthood often lack specialists in their rare diseases. Either those specialists are difficult to find or they don't even exist. Currently in the state of Minnesota, there are rare disease patients who are reaching age 20 and 21 who, as they transition out of their pediatric services, have no providers to go to at all. And this is a huge barrier to care. If a rare disease patient is fortunate to have a specialist, he or she, that specialist might be located in a geographic region miles away, or they might be in a different state, and sometimes even being able to access care across state lines is a huge challenge for the patient population. The Council will work to identify solutions to these barriers, such as providing lists of providers with expertise in various diseases and better educating the larger medical community on the lack of providers, so that this becomes a priority in the medical system. Pillar number 3 is reduce time to diagnosis. And the goal of the Rare Disease Advisory Council will be to advise, consult, and cooperate with multiple institutions in the state to develop information and programs that increase awareness for the diagnosis and treatment of rare diseases. The average wait time to a diagnosis for a rare patient is 6 to 7 years. I've met people that have waited far longer to receive a diagnosis for their rare disease. Additionally, a rare disease patient is misdiagnosed on an average 2 to 3 times. This delay in diagnosis creates inefficiencies in the system and significantly negatively impacts the patient's quality of life and health. The Council believes that the starting point of reducing time to diagnosis is more awareness of the need of the medical community to consider the fact that a patient may be living with a rare disease, as well as an awareness of where that provider can go for resources. So we will seek as a Council to be that informational resource for providers and patients. And pillar number 4, which is our last pillar, is the acceleration of research. Now, the term acceleration of research can take many different forms, and the Minnesota Rare Disease Advisory Council will foster the increase of rare disease research through awareness and collaboration. Some of you in this room are future researchers, and the Council will encourage the scientific community to consider prioritizing research that advances care for rare patients. Again, with the knowledge that rare is not so rare, collectively rare disease patients affect 1 in 10 of the population. In addition, by fostering the growth and maturity of disease-specific communities, these communities these patient communities can then support the scientific communities. I often say that scientific advancement is often predicated on a healthy and functioning patient advocacy community. such things, patients can do things such as work to organize and build their community so that clinical people can be identified to participate in clinical trials quickly. Patient communities can form patient registries and conduct conferences that greatly educate the scientific community. Patient communities can conduct natural history studies to give the much needed information that's lacking to the scientific community, and this accelerates research. As I've said before, currently only roughly 5% of the 7,000 rare disease patient populations have an FDA-approved treatment for their specific disease, and I don't even think I need to comment on that statistic. I think we can all agree in this room that that is an unacceptable statistic, that we need more research. Now that we've articulated our vision and our priorities, these priorities will inform our path forward and allow us to fulfill our commitment to the state of Minnesota and to the patients who work so hard to make this Rare Disease Advisory Council a reality. Thank you so much, and I'm going to now turn this over to the next speaker.
Hello, good afternoon, everyone. My name is Brenda Ogle, and I am the current director of the Stem Cell Institute here at the University of Minnesota. And I have the wonderful job of introducing our keynote speaker for today. But before I do, I thought I would just share with you a little bit about the Stem Cell Institute and why we're so excited to partner on this event today, as we have for the past several years. You heard Dean Tolar speak earlier, and I echo his sentiments. He was actually the former director of the Stem Cell Institute and began this partnership to participate in this day. So the mission of the Stem Cell Institute overlaps with much of what you are interested in accomplishing as well, and that is to advance human health. And our institute is driven by, or was driven by, the isolation of stem cells from the body. And our goal is to conduct research to better understand how stem cells operate in that space, how we can control their behaviors or how those behaviors are controlled, and then how we can use them to advance human health in at least, well, several ways, but two of the top two ways are to first develop model systems outside of the body, models that mimic human tissue. And that way we can better understand disease progression. We can test therapeutics before they're used in people. The second is that we hope those models and other studies lead to the development of therapeutics. And I think it's exciting in many ways. You know, a lot of this research started by focusing on the most prevalent diseases in our society, or what we perceive to be the most prevalent. As you heard earlier, we are a heterogeneous group of people. However, all of those diseases, because stem cells can be generated from individuals, from mature cells of individuals now, we can start to use those models to develop patient-specific models, and that's where we start to get at rare disease. We can also use gene editing approaches to impose the mutations that might be associated with rare disease. So that's what we're doing in a nutshell at the Stem Cell Institute, and we would love to have engagement or interaction with all of you. Today is one of those special days that we have that opportunity. In the next few months, in September, I'd like to point you to another opportunity to engage with us at the Stem Cell Institute. And that is part of our 20th anniversary celebration. We've actually been in existence for 20 years. We were the first institute in the nation dedicated to stem cell research. We're quite proud of that. And one of the things that we're doing to celebrate on this day is to hold a community forum that we're calling a Lunch and Learn. So you're all invited. I invite you to check out our website for details as they emerge. And the concept is to talk about or have a discussion about the present and future of stem cell therapies. Something we're asked pretty routinely in the Stem Cell Institute is there any stem cell therapy out there right now that's safe or effective? There are so many that are kind of bubbling to the surface. So we're going to discuss that. We're bringing in several speakers from the outside, including George Daly, who heads up the Harvard Stem Cell Institute, as well as many of our strong faculty. After a discussion, they're going to sit on a panel to take questions from the community, and they will include members from industry who are also focused on stem cell therapies, and also members of the legislature. So I invite you to come and join us, and we'd love to engage more with all of you. So with that, I'd like to introduce our keynote speaker, Sarah Wicks. She comes to us today. She's an associate at Hyman, Phelps, and McNamara. She had her training at first with a bachelor's degree in biology, followed by a master's of public health from Johns Hopkins University, and finally her Juris Doctor from the University of Maryland. She works very steadily to help many sponsors bring their therapies through pre- and post-market FDA landscape. She helps them navigate that, but she specializes in those sponsors who are looking to treat rare disease. And most uniquely, she's trying to incorporate the patient perspective and experience to more effectively move these therapies forward, or at least determine more quickly if they will be effective. So with that, please help me welcome Sarah Wicks.
So thank you very much for that kind introduction, and thank you all for having me here as we celebrate Rare Disease Day and World Rare Disease Week. I think it's fitting that we talked about the Rare Disease Council and also how everyone is just really working together to help propel not only the industry forward, but also how FDA is thinking about helping the patient community and rare disease patients. So with that, I'm going to start out with talking about FDA's gold standard for approval and how the Orphan Drug Act plays into the approval of rare disease therapies. Also, and most importantly, what FDA's flexibility is in approving rare disease therapies and how the patient's voice plays a role in that. So starting at the beginning with the 1962 amendment to the Food, Drug, and Cosmetic Act, this is really where we see the gold standard of approval codified by Congress. The 1962 amendments created requirements for pre-approval of all new drugs, to include the requirement of substantial evidence consisting of adequate and well-controlled investigations, such that the FDA can conclude that the drug will have the effect that it claims to have in its proposed labeling. And FDA's general interpretation of this 1962 amendment has become known as the gold standard of approval, a minimum of 2 adequate and well-controlled clinical studies, where each trial meets its pre-specified primary endpoint in a statistically significant way. So we know that 10% of Americans are affected with a rare disease. There are more than 7,000 rare diseases identified, many that we don't even know about. 30 million people are affected collectively with a rare disease. Yet, early on, companies were not developing drugs to treat rare diseases, and there's just inherent challenges in doing such research and bringing therapies to market. The population is so small that oftentimes it's difficult to even power a study to show a clinical effect. Disease pathophysiology and natural history is often poorly understood, if understood at all. And there's a question about what endpoints are actually should be looked at when conducting such studies. And the result of these challenges was essentially a small commercial market with less than optimal knowledge to guide clinical development, and certain aspects of just drug development still being considered infeasible. And this is where the Orphan Drug Act comes into play. So in 1983, President Ronald Reagan signed the Orphan Drug Act into law, which created a number of incentives for drug sponsors to engage in research and approval of these therapies for orphan drug development. These incentives included the availability of grants, tax credits, and 7-year market exclusivity. And since the passage of the ODA, we have more than 500 orphan products approved for more than 800 rare disease indications. 10 years prior to the Orphan Drug Act, only 10 products had been approved for rare diseases. So this is a really immense accomplishment. In 2019, 22 of the 53 novel drug products approved by FDA were drugs to treat rare diseases. And the year prior to that, in 2018, we saw the largest number of rare disease therapies approved by the FDA ever. So moving on to how FDA has flexibility in approving orphan therapies. So FDA has always had, aside from the gold standard from the 1962 amendments, FDA has always had the authority to exercise flexibility in its regulations. Specifically, under this regulation, 21 CFR 314, while the statutory standards apply to all drugs, the many kinds of drugs that are subject to the statutory standards, the 1962 gold standard, and the wide range of uses for those drugs demand flexibility in applying the standards. Thus, FDA is required to exercise its scientific judgment to determine the kind and quantity of data and information an applicant is required to provide for a particular drug to meet those statutory requirements. So moving on to the types of flexibility that FDA has, I first want to start with formal policies that were either designed by Congress or by FDA to meet the standard beyond the 1962 standard so that they could formally exercise flexibility. And I'm going to start chronologically. Accelerated approval. This system was created in the 1980s in response to the need of patients contracting HIV infections and the attendant public health crisis. And this program was placed into statute by Congress in the FDA Safety and Innovation Act officially in July 2012. Prior to that, FDA had just been using this program on its own. So accelerated approval is a program, an approval pathway by which a therapy for a serious or life-threatening disease that generally provides a meaningful advantage over available therapies, if there are any, may be approved based on a surrogate endpoint that is reasonably likely to predict ultimate clinical benefit. And subsequently, because this approval is based on a surrogate endpoint, sponsors that have used this pathway must engage in a post-approval requirement study to establish that that surrogate endpoint ultimately led to the clinical outcome benefit. In 1997, the Food, Drug, and Administration Modernization Act, known as FDAMA, 115 codified FDA to consider data from one adequate and well-controlled clinical investigation and confirmatory evidence. And for a while, we didn't really know what this confirmatory evidence meant. And we also didn't know how robust an adequate and well-controlled clinical investigation had to be if we could use confirmatory evidence. So very recently, actually, 2 months ago, December 2019, FDA came out with a formal policy, a guidance document that actually lays out what that confirmatory evidence is. And this very detailed way describes what types of evidence constitute this confirmatory evidence. And it can be results from trials in a related indication, such as 2 or more completed adequate and well-controlled trials demonstrating efficacy in an indication where FDA may accept one trial in a related indication that uses a similar mechanism of action in producing clinical benefit. Compelling mechanistic information from earlier clinical or non-clinical studies, such as reliance on a pharmacodynamic endpoint with well-established relationship to the clinical endpoint, and reliance on well-established animal models. Also using well-described natural history of disease, such as evidence that clearly describes the natural history through a registry or compelling case series or a natural history study. And lastly, an adequate and well-controlled trial from another member of the same drug class, such as the same pharmacologic target. And also, we have the May 1998 guidance document providing clinical evidence of effectiveness, which allows FDA to approve a therapy based on a single, adequate, and well-controlled study without independent substantiation. So, FDAMA 115, a single, adequate, and well-controlled study with confirmatory evidence. May 1998, single study, and you're done. But back to that, sorry. I just want to go through that this guidance does lay out what would constitute a single and adequate, well-controlled study. And these characteristics are the same characteristics that you would expect from the 1962 gold standard, including a large multi-center study, consistency across study subsets, multiple studies inside a single study, such as a factorial design, multiple endpoints involving different events, and then also a statistically very persuasive finding. So this is a very, very low p-value. And not one of those characteristics is necessarily determinative, but the presence of one or more of those in a single study can help contribute to the conclusion that the study would be adequate to support an approval. So members of my firm have done an analysis of FDA's flexibility and essentially looking at all orphan drug approvals since 1983 and determining whether FDA had exercised some form of flexibility in those approvals. So the scope, we reviewed all orphan drug new chemical entities, excluding those for rare cancers, just because those are usually their own animal. And the first analysis, published in 2012, looked at approvals from the ODA 1983 to 2010, and 135 approvals were looked at. From July 2010 through June 2014, 27 approvals were looked at. And we are currently in the process of updating the analysis for a third time for the approvals from July 2014 through to June 2019, and we are looking at 58 novel orphan drug approvals during that time period. And for each approval, FDA publishes on its website all of the review documents from the review process, and these are publicly available on its website. And we primarily looked at the medical and statistical reviews, the director memos, cross-discipline team leader memos, and classified the level of efficacy evidence that was determined by FDA to be adequate for drug approval. So each approval was classified into 3 classes of efficacy evidence. The first being the conventional gold standard of approval as outlined in the 1962 amendment to the FDCA. The second bucket is under administrative flexibility based on formal FDA policy. And this includes the May 1998 guidance, single study approval, FDAMA 115, single study approval with confirmatory evidence, and accelerated approval. And then if an approval, didn't fit nicely into any one of those buckets, we called it an ad hoc case-by-case review. So the results of the 2012 analysis were originally published in the DIA journal, Drug Information Journal, and out of the 135 approvals reviewed, 90 of those approvals had exercised some form of flexibility on the part of FDA. For the 2015 update, 19 of the 27 orphan drug approvals resulted in some form of exercise of flexibility by FDA. And so far, in the 2020 analysis, out of the 53 of 58 approvals that we have looked at, 34 of those 53 approvals have resulted in some form of flexibility. So this is really common. FDA is finding ways to bring therapies to the rare disease community. The gold standard 1962 standard is not feasible when assessing these types of therapies, so FDA is really utilizing these other flexible authorities to bring these therapies to market. So just to go further into the breakdown so that you can see, we had, out of the 34 approvals that used flexibility, we had 26 that were based on either the May 1998 guidance, FDAMA 115, or accelerated approval, and 8 were based on case-by-case flexibility. And looking even deeper into the 2020 analysis, we see that 8 approvals were based on accelerated approval, 11 were based on a single adequate and well-controlled study that was highly persuasive, highly statistically significant. 7 of the approvals had a single study approval with confirmatory evidence, and 8 were based on a case-by-case analysis. So I really just want to go through an example of what each of those types of flexible approvals looks like. The first being accelerated approval. Here we have Exondys 51 eteplirsen for Duchenne muscular dystrophy. DMD is an X-linked recessive neuromuscular disorder caused by mutations of the dystrophin gene, which disrupt the mRNA reading frame, leading to an absence of dystrophin. And this absence of dystrophin leads to muscle damage, loss of physical function, and ultimately premature death from respiratory or cardiac failure in the 2nd to 4th decade of life. And at the time that this product was submitted for review, there were no FDA-approved therapies at the time of submission. So the mechanism of action for eteplirsen is that it was designed to restore the mRNA reading frame of the dystrophin gene, but it was not a complete restoration. It resulted in a truncated form of the dystrophin protein, and this truncated dystrophin protein is actually found in a less severe form of muscular dystrophy called Becker muscular dystrophy. So when reviewing this submission, the question wasn't whether dystrophin level is an appropriate surrogate endpoint. The question was whether the quantity of dystrophin produced by the therapy had an effect that was reasonably likely to predict clinical benefit. Because remember, for an accelerated approval, you're basing the approval on a surrogate endpoint that is reasonably likely to predict clinical outcome. So here, dystrophin is obviously an appropriate surrogate endpoint. The question is how much dystrophin needs to be produced by the therapy to create a clinical benefit in patients. So Dr. Woodcock, the director of CEDAR, and Dr. Unger, the director of the Office of New Drugs Evaluation, They looked at the submission and they agreed that eteplirsen in a randomized controlled trial produced statistically significant increases of dystrophin compared to control. However, the level of dystrophin that was produced did not rise to the levels of dystrophin that's seen in Becker's muscular dystrophy, which is the lesser form of muscular dystrophy. So the question was, is that enough dystrophin to have a clinical meaningful input on patients that have a more severe form of muscular dystrophy? And there were some discussions about the natural history and disease progression of muscular dystrophy, particularly DMD. And it was ultimately determined that in these patients where there is such heterogeneity across patients in this one disease, that there is no rational basis for identifying a threshold level of dystrophin levels. So any increase in dystrophin level would be considered a clinically meaningful benefit in these patients. An example of a FDAMA 115 approval, again, FDAMA 115 being a single adequate and well-controlled trial with confirmatory evidence. Study CS3B was the single adequate and well-controlled study. It was multi-centered, it was randomized, double-blind, sham control, all the things we like to see in an adequate and well-controlled study. The primary efficacy endpoint was the proportion of motor milestone responders, and this included effect on voluntary grasp, the ability to kick, head control, rolling, sitting, standing, and so forth. And this study was extremely statistically significant. 21 out of the 51 patients on drug achieved a motor milestone response. 0 out of 27 achieved a response in the sham control. And there was also supportive evidence from the secondary endpoints that further drew this conclusion. So it probably could have stood on its own, but the sponsor also applied with 6 open label studies. And these trials were done in different subsets of the population with SMA. 4 trials were done in patients with later onset SMA. 1 trial was done in pre-symptomatic infants, and 1, again, in the infantile onset SMA population. And in all of these studies, major milestones were achieved, and ultimately this allowed the product, the therapy, to have an indication that did not restrict age or of onset or the subtype of SMA, even though in the first study that was conducted that was highly statistically significant, that was only done in infantile onset SMA. So using these additional open label studies, they were able to get an approval that covered all types of SMA. Moving on to an example of a May 1998 approval, which is again is based on 1 adequate and well-controlled trial that is highly statistically significant. I chose Radicava for ALS. So this is a very unique example. I dove deeply into this product for a client question, so I know a little bit about it. So the first study that was performed was Study 16, and it was a 24-week double-blind placebo trial, meets the adequate and well-controlled standard. Primary efficacy and endpoint was the ALSFRS R scale, which looked at fine motor, gross motor, bulbar, and respiratory functions. Now the p-value, this would be considered a failed study based on the p-value of 0.41. And the sponsor decided to do a post hoc analysis and looked back at the population that they had recruited. And they actually identified a subgroup of patients that they thought had the potential to be more responsive. And based on this subgroup of patients, they identified them as having a better functional status at baseline. They had a higher forced vital capacity, and they had been diagnosed with ALS within 2 years. So based on all of that information, the sponsor went back and did another study, study 19. Same exact design as the first study, but in this new subgroup of patients. And here the result is amazing. They had a highly statistically significant endpoint. Just by looking at a different subset of patients in this population. So we had 2 studies, and one was a failed, adequate, and well-controlled study. One was a highly statistically positive study. So here we were really deciding what category did this fall into? Was this a FDAMA 115 approval, or is this a May 1998 approval? It's based on a single study with no confirmatory evidence. Was study 16 confirmatory? And here, looking back at the review, looking at what FDA thought, they all clearly agreed that study 19 was highly robust, could stand on its own. The question was about study 16. And here, FDA essentially thought that study 16 was merely hypothesis generating. Without study 16, they would have never identified the subgroup of patients to further study in study 19. So you can learn a lot from looking at the review of a therapy. And lastly, I want to talk about an example of an ad hoc case-by-case approval. I will note that this is a very unique approval just based on the number of patients alone. So Zuridine, uridine triacetate for hereditary orotic aciduria. The sponsor submitted 2 clinical sources, the first being 19 case reports that had been published over the course of several decades, none of which the sponsor had obtained themselves directly. And then also the results of a clinical trial that included 4 patients. So I will say that HOA would be considered an ultra-rare disease. There are about only 20 patients identified worldwide with this disease. So you would never be able to do an adequate and well-controlled trial in this particular disease. So based on this 6-week single-arm open-label study in 4 patients, 3 of whom actually had already been treated off-label with uridine. Based on this study and an extension study of up to 6 months, the sponsor conducted the study. So all of these things, in addition to there not being one single primary efficacy outcome, each patient had their own clinical outcome that was measured separately based on their unique manifestation of the disease, 3 of which I have listed here, such as neutrophil count, total white blood cell count, and red blood cell mean corpuscular volume. So some patients had already previously been treated with the therapy off-label, and then each patient had its own clinical endpoint that was measured. So this is clearly a case-by-case flexibility on the part of FDA. So moving on to the evolution of the advocacy piece and the importance of the patient voice. The beginnings of patient advocacy at the FDA really started in the 1970s and 1980s. The Women's Breast Cancer Advisory Center in 1964, started by Rose Kushner, and also the AIDS epidemic really was the beginnings of patient advocacy, where patients were literally on the steps of FDA demanding that their needs be met. Today, we have thousands of representative organizations in the United States that serve as policy and funding advocates. They do outreach and education, not only for members of their patient group, but also to researchers and academics in the community, and really work hard to bring the patient perspective to the community. So the role of patients was a very, very long time coming. The nation's first law on medicine was the Pure Food and Drug Act, passed in 1902. And as I had mentioned earlier, the Food and Drug Administration Safety and Innovation Act, which codified the accelerated approval pathway in 2012, states that to develop and implement strategies to solicit the views of patients during the medical product development process and consider the perspectives of patients during regulatory discussions. Prior to this 2012 law, patients had never been mentioned in FDA lawmaking by Congress, in FDA regulations. So this was 110 years in the making for patients to officially be recognized by Congress and by FDA in their policies. And now we have the 21st Century Cures Act, specifically Section 3001, which requires the FDA to state the patient experience data that they have relied on or have submitted, had reviewed as part of an NDA or a BLA. And this is an example of one of the first drugs approved with a dedicated patient experience data section. And this is something that is becoming, it's required now, so this should be more mainstream now in all of the approvals that are happening as we move forward. And now we also have patient-focused drug development meetings. PDUFA 5 includes dedicated resources to expand activities that will provide the review divisions with patient input. Since 2012, FDA has convened 25 meetings, both in disease areas that are more conventional diseases and also rare diseases, in an attempt to look at the armamentarium for that indication and also identify areas of unmet need. And participants at these patient-focused drug development meetings include FDA review staff, relevant patient advocacy communities, and other interested parties such as sponsors. And now, since December 2015, we are seeing that not only is FDA hosting these PFDDs, but patient advocacy groups can now host them themselves as well. And these are called externally led PFDD meetings. So since December 2015, CDER announced that these meetings can actually be conducted by patient groups and hosted by those groups. Patient groups submit a letter of intent to the agency, putting the agency on notice that this meeting is going to happen. FDA is invited, FDA comes to the meetings. But otherwise, the patient group is really responsible for running the meeting, deciding what is going to be discussed at the meeting, and things of that nature. And most importantly, the voice of the patient report is something that the patient group puts together, and this is submitted to the FDA so that when they have a therapy come through their review division, they can reference this voice of the patient report to really assess the benefits and the risks of a particular therapy that the patient group has identified. So why is patient voice key to orphan drug development? FDA decisions are often less clear-cut, as we saw with my examples earlier this afternoon. And based on the challenges of developing therapies in the orphan drug space generally, we have small, patient community sizes that can engage in the clinical trial process. They're dispersed geographically across the country. We don't know what efficacy endpoints to look at because we don't fully understand the disease. And oftentimes it's infeasible and unethical to not only conduct a second study in this patient group, but there's even a question of whether you can do, if you can have a control. If there's no current available therapy for these patients, why would you not want everyone on drug in a clinical trial? So there are inherent challenges, but patients can share their experiences and preferences that can help identify the burden of the condition and the unmet medical need, what constitutes clinically meaningful treatment benefit, risk tolerance compared to the benefits of the therapy, and other aspects of a product that improve patient care, such as the route of administration. And with that, I just want to say thank you, and I welcome any questions.
So you've heard today about the remarkable evolution that's taking place both nationally and here in our state. So here's an opportunity for those of you in the audience to offer comments or ask questions. And we have someone, or did. Is there a microphone roving around? Yes, two of our highly skilled students, 3 of our students are going to be able to carry around microphones so that you may either ask a question or offer a comment. Anybody to begin? Okay.
So I had a question on the ALS study with Radicava. So what was it about subgroup or study 16 that allowed them to gain insight into as to what subgroup would then be used for study 19?
Sure. So as a post hoc analysis, they looked at all the data, all of the inclusion criteria, and it was really just a matter of identifying patterns in the patients that had conducted the study, and trying to find a pattern, if any, to inform another study since the first study had failed. And I think this is a good lesson for sponsors, that this is a failed study can still be informative for the next study. And even if study 16 had been a little bit more successful, your confirmatory evidence study does not have to be the second study you conduct. You can use the first study to confirm a very successful second study as well.
Could you comment on what those patterns were that have to do with their forced vital capacity, or what were the markers that led them to say these are the patients that we could now enroll with greater signal to noise?
Yes, so they looked at forced vital capacity, identifying patients that had a VVC greater than 80%. They looked at how severe the disease was in certain patients, identifying those patients with more severe disease as being, or less severe, as potentially being more responsive. And then the earlier the diagnosis of ALS as also being a potential marker. So as far as how scientific the diagnosis piece is, I'm not sure, but those are some of the things that the sponsor had looked at in identifying the subpopulation of potential increase in responders.
Thanks. Hi, I'm Shelley. I'm a genetic counselor, and this question is for Erica and Sarah. Huge news story last year was the N of 1 drug with the young girl, Mila. I'm wondering to me, that felt like a huge leap, and I'm wondering what kind of conversations you're hearing or thoughts on this big change, I guess, in approval of drugs.
So we're seeing a really dramatic shift, and I think my last case-by-case example with the Zuridin approval based only on actually a clinical study in 4 patients is becoming it's the challenge is more identifying sponsors that are willing to conduct those studies. But as sponsors become more invested in learning who the patient community is, we're seeing this more and more, and especially in the gene therapy realm where it is N of 1 based patient per patient. It's extremely popular and exciting to be in this space now for those reasons.
Patty.
Thank you for a really interesting presentation. It clearly is obvious that over time the agency has statutory basis for being more flexible. My question has to do with the approval of Exondys. You know, there was, you described it as one of your cases in the agency using regulatory flexibility, but we also know that there was a lot of controversy around that. And, you know, are there things afoot, any activities afoot to help people within the agency get used to some of this regulatory and these legislative changes, so once some of this flexibility is utilized, there isn't this same kind of consternation, if you will, within the agency?
So we are seeing a lot of, unfortunately, heterogeneity across review divisions within the FDA on what they deem sufficient levels of efficacy in making their benefit risk determinations for approvals in the rare disease space. And one way that by we I mean everyone in that we work with, as far as industry and the patient community, lobbying, I would hate to use that word, but we are advocating for a rare disease center of excellence of sorts, which would be a way to harmonize the way that rare disease drug applications are reviewed at the agency, so that people who have experience in the science of small trials, who have experience as clinicians in treating rare diseases, are housed within one center at FDA to really harmonize the views and the way that these applications are approved. We do have, working with sponsors, we do have an idea of the offices and divisions that will give us questions and really make us prove our case. So unfortunately, that is a real thing, and we are trying to figure out how to harmonize that effort.
Yeah, good afternoon. I'm Spencer Merchant. I work at United Healthcare, and question for you, Erica, and your council, and I applaud all the work you've been doing. You have a lot of people that are rooting for you at my company, and can you describe the breakout and the kind of makeup of the council? You mentioned about 20 or so members, and sure. Even in your sort of early formatory time, how are you kind of getting everyone to beat to the same drum and drive to kind of that mission?
Yeah, that's a super question. So to speak to the makeup of the council, it's quite large. There's 24 members on the council, and as we started to build out the legislation, we really had a strong desire to have all the stakeholders, representation from all stakeholders across the spectrum. So there's a payer on the council, industries represented, the patient advocates, researchers, doctors, genetic counseling, and social work, and legislators. So really trying to bring multiple perspectives. Our thought process was the more perspectives that we have, the better. In terms of getting everybody to kind of march to the same drum, that's always a challenge with any effort. And it probably sounds very, very trite, but it's absolutely true. You know, really, there's three of us that represent patients and the patient community on the council. So there's three positions for patient groups, and we always try to bring it back to how is the work we're doing directly going to affect the patients that we see, and how is this directly going to affect patient lives. So I don't think there's anything more powerful than having a group of people in the room who have a lived experience with rare diseases. And then on pragmatic scale, trying to find goal alignment with things we can all agree on. The exciting thing is there does seem to be a growing recognition in society and across all sectors that the barriers are problems that we're all responsible to solve. So there does also just seem to be a sense of urgency. Every time the work groups get together, some of the council members are in this room, the work groups they bring a sense of urgency to the conversation every time we get together. So it's very exciting.
Erica, along those lines, several people approached me and they asked how could they, either as patients or members of advocacy organizations, interact with the council. Can you respond?
Sure, that's exciting. And for the last 3 months, we've really just tried to to operationalize the council, get the procedures established. But in the next couple of weeks, we're going to actually launch a website. And there's a way to sign up for a newsletter on that website. And I am always available. If you see me, come and see me. I can give you my email address. I want to hear from patient groups. I want to hear from stakeholders and get your feedback. We're also we're in the process of designing some surveys, patient surveys as well as physician surveys, and not just physicians, but care provider surveys. So that's a way we're going to get feedback from the community. So.
Other questions or comments for our panelists?
This is another question for Erica. Do you know of any efforts like yours going on in other states as well? So is there any kind of template or corresponding organization?
So in fact, North Carolina passed the first Rare Disease Advisory Council in, I believe, 2015. And it was actually, I had read about that council, and that was, at that time there was only one. Since then, I believe we're up to 7 states have rare disease advisory councils, and they all conceptualize it a little differently based on their own state's dynamics. But there's about 7 states. The National Organization for Rare Disorders kind of is helping with the effort and kind of overseeing that. So we actually are setting up regular times to communicate with each other. So it is kind of a phenomenon that's taking off around the state, but Minnesota's is the best. So I just really think that objectively.
While we're waiting, and this is to Sarah. Sarah, with the era of gene therapy, we're now seeing trials in very rare disorders where the studies may begin with an N of 1 and may have a total number fewer than 10. Can you share any information you have about how the FDA is going to look at these sorts of therapies?
So I think the agency's current focus with gene therapy right now is not so much on the clinical trial side. They have plenty of experience in engaging with sponsors and designing trials before they even get to the beginning points of a trial. The agency works really well with sponsors in the design of trials, and specifically with gene therapies, there are special routes within the agency for sponsors to engage with the FDA. I think what we're seeing is that the agency is more concerned with the scaling of the drug product to the stage where that will be manufactured on a larger scale for the clinical trials and how they maintain the integrity of the product.
Hi, my name is Mary Bearden, and I'm being gene therapied right now, so I'd like to have a second gene therapy, and I understand that there's always phases they have to go through, but I'd also heard that there's the new antisense that's going through the 3rd phase right now. It's going to start in June, that they might speed that up because that will change the lipids on the lipoprotein A, which hadn't been able to be treated before without apheresis, which kind of is the other cardiovascular disease. Do you know anything about what's the, for the patients that can't get in the trials, we'd like to have that 3rd phase sped up. What's the criteria for that? I think it's at a 5-year right now, and we were hoping it would be 1 year.
So on the sponsor side, there's always recruitment issues and enrollment issues, especially for rare diseases, identifying patients who are spread out geographically, so that can always hinder the progression of a clinical trial, especially a phase 3 trial, which generally are larger in size than phase 1 and 2 studies. But I haven't worked directly with gene therapies yet. We do at our firm, but those are just some thoughts on what could be hindering the progression of the phase 3 study. As far as interactions with FDA, at the stage of phase 3, the sponsor has communicated with FDA previously, so there's probably potentially a scale-up issue, as I had mentioned, with the chemistry, manufacturing, and controls, making sure that the stability of the product is efficient, sufficient, but also from a public health standpoint, that's FDA's role is to further the public health. So if they could speed up the phase 3, they would. We're seeing that done in other therapies, especially in medical countermeasure and tropical and infectious diseases. We see products move a little bit faster just because there is an impending public health crisis. So if the agency could do it, they would.
Well, with that, I'd like to thank both Sarah and Erica for some informative presentations, and join me in showing your appreciation. It's now time to celebrate the success and the creativity and ingenuity of the next generation of researchers. And those are the recipients of our awards program that go to trainees across the state, actually. And as I do that, I just wanted to point out, and it occurred to me as I was walking around, that we have in this audience people from high school to those of us who represent an older generation. And it really speaks well to the vitality, diversity, and the robustness of this group all coming together. And so the awards are made possible through a generous contribution from the Chloe Barnes Rare Disease Foundation, which is represented here by Erica. We have 4 recipients, 3 of whom are able to join us today, and I'd ask them to come to the stage. So the 4 recipients, and these were judged by an interdisciplinary panel of faculty, are listed here. And as I go down this list, I will bestow an award and then a certificate, a gift certificate. And is Dr. Argo here? Oh, okay. Could you join us as well? Talk about surrogate endpoints. Dr. Argo is a co-author on one of the posters by Colin Gardner, and we want to recognize him. He was unable to join us as well. The first recipient is Abhishek Sate in the Department of Experimental and Clinical Pharmacology, and he presented a poster on the fact that across the nation we're giving lower than appropriate doses to treat a serious rare condition called status epilepticus, and hopefully through his work we'll begin to raise the awareness of that particular clinical problem and improve outcomes. So Abhishek, here you go, and here's the certificate. Congratulations. Our next recipient is Julie Riedel. Riedel. Thank you. I had a tough time with that. She was explaining this is a German origin name, is that right? Yeah. And she was going into the differentiation between vowels in German language, which was very helpful to me. And she's doing some fascinating work on a new approach to looking at the possible stem cell therapies or cell therapies for this rare disorder, epidermolysis bullosus. And so we congratulate her on her work, and we hope she continues to proceed with it. Here you go. And we'll do a photo op here. And your gift certificate. Thank you. And last, Dr. Argo, please join me. And she's representing Cullen Garden, who is a third-year medical student, and his work was on really a remarkable case report, which just exemplifies what rare disease challenges are, and looking at the findings and identifying a particular type of a rare disorder. And so with that, I want to congratulate their team, and on behalf of the Orphan Day program, congratulate you and your co-workers. Dr. Gottner could not be with us today, and so we will present him with his certificate and his award at a later date. And I want to again thank the recipients. Best of luck. We look forward to your future contributions. It's now time to bring our program to a close, and on behalf of Brenda Ogle and the Center for Institute for Stem Cell Research, Stem Cell Institute, and the Center for Orphan Drug Research, we are so pleased to offer this program. And if we could move to the next slide. And you've heard a little bit about the Stem Cell Institute, and just a brief word about the Center for Orphan Drug Research. We were founded in 2005. Our focus is a mission to develop new therapies, whether they be new drugs or old drugs that are repurposed, but dedicated to rare disorders. We have a mission to educate, and educate at all levels within the healthcare system and outside the healthcare system. And we have a mission to promote rare disease advocacy and understanding in the community, and this program is an example of that effort. Our website is down below, and we will have the slides posted there if you want to go back and get additional information. And we'll work with Erica to make it also easy for you to connect with the Rare Disease Advisory Council. And if I could go to the next slide. Now it's time to do some thank yous. These are our sponsors. We couldn't do this without the help of those who are willing to contribute to the program. Many of them were here today. If you hadn't had a chance, and they still are around, we would ask you to convey your appreciation for their support. The next slide, please. And this is the remarkable list of patient advocacy organizations. And one of the things I heard over the course of the day is that one group wasn't aware of what another group was doing, and they learned more by just being here, and we hope that continues. That's an important outcome of this program. And again, working with the Rare Disease Advisory Council, we hope to be able to promote more of these interactions. Next slide. And then the Planning Committee, who worked very hard over the last 12 months to put this program together. They're listed here. You saw some of them today. And I want to offer a special thanks to Lori Inslee. Lori, will you please stand up? Lori is the administrator for the Center for Orphan Drug Research and has been really just instrumental in putting this program together while in part on maternity leave, and was able to come back and pull this together in a timely and a very effective way. So we offer special thanks to you, Lori. Next slide. What are we about? This program is about providing information, and it's about providing inspiration, and it's about offering hope. And I hope during the course of the day, all three have occurred. And I can guarantee you that we will continue this effort. So join us in 2021, information to follow, so we can continue our journey along the path of improving the care of those with rare disorders. Thank you all. Program is over.